Hello everyone, my name is Zara Qasemi. Today we will go over dialysis technique and also we will watch a lab demonstration. As you recall, in diffusion, molecules or substances move down their concentration gradient. The solute diffusion across the selectively permeable membrane is called dialysis. The dialysis technique was introduced by Thomas Graham in 1861, and scientists use an artificial cell that is called dialysis tubing for many purposes, such as purifying substances like drugs. It has some macroscopic pores that we cannot see it with the naked eye. So this tube works as a selectively permeable membrane. It would allow some molecules smaller than these pores across the membrane, and any substances or molecules bigger than these pores would stay in the bag. This is a dialysis tubing. If you don't know how to assemble the dialysis tubing, make sure to click on the link provided in the description. So I fill this bag with phenolphthalein and water. Phenolphthalein is a pH indicator and it turns pink in a basic solution and colorless in an acidic or neutral pH. In the beginning, as you see here, inside of the bag was colorless and filled with water and phenolphthalein. And outside of the bag, I poured NaOH, that is a stearin base. After 20 minutes pass, as you see here, inside and outside turn to a pink color. The pink color of outside of bag indicate the presence of the phenolphthalein. That means phenolphthalein molecules diffuse through the membrane and turn the outside solution pink. At the same time, as you see here, the inside of the bag turn to pink, and the molecules that diffuse to the bag were NaOH molecule. That's why the inside also turned pink. Thank you for watching this video. I hope you find it helpful. I would like to encourage you to watch next video, that is the lab demonstration over osmosis in an artificial cell. Stay tuned.